Good afternoon, everyone. Thank you very much for joining us at the I3L's Power Talk today. I'm very happy to see that there are a lot of participants, even though today is actually a public holiday. So happy Islamic New Year to all of you who celebrate it. I hope you're happy, you're healthy and safe at home. For today's topic, we have a very enticing title here, the Microbiota-Gut-Brain Axis, Hype or Revolution. We're very happy to have Professor Dr. Lukas Van Oudenhove, MD, PhD, from the KU Leuven in Belgium. Good morning again, Professor. Nice to have you here.
Thank you.
Next slide, please. Thank you, Wirda. So for the agenda for today, it's going to be around a 1 hour session. After the opening, we'll go directly into our speaker's presentation for 40 to 45 minutes, and we will follow it up with a Q&A session. There will also be an evaluation session, so Wirda will help us send a link, and please fill it in. So this is for us to review and always improve our power talks. It will also be useful for you because you will get an attendance certificate afterwards. Next, please. Yes. So before we start again, also a bit of some housekeeping rules. So for all attendees, please don't activate your audio during the Power Talk session. Keep your mics muted so that we don't disrupt the talk. But I do encourage everyone to use the chat feature to ask questions. You can do this throughout the chat, throughout the talk, or afterwards you can also open your mic if you want to ask questions directly. Like I mentioned earlier, there will be evaluation surveys distributed to you at the end of the event. It's a pleasure for me to be able to introduce Professor Lukas Van Oudenhove today. So Professor Van Oudenhove is a psychiatrist and also a researcher in the field of gut-brain axis at the KU Leuven. His position now is an associate professor at the Faculty of Medicine at the KU Leuven. He's the principal investigator of LaBGAS, Laboratory for Brain-Gut Axis Studies. He's also the member of LBI, or the KU Leuven Brain Institute, and the member of Leuven AI, or the KU Leuven Institute for Artificial Intelligence. He has been awarded numerous research grants to fund his ongoing projects. Now he has Mood Bugs, which is about how gut microbes influence our emotions. There's also From Pavlov to Visceroception, about the effect of fear learning on perceptual habituation and discrimination of visceral sensations. And there's also a project about satiation and reward without the calories. These are not his only projects. There are several others. So if you're interested, please, you may have a look at the LaBGAS website, which is written down below, gbiomed.kuleuven.be. Professor Van Oudenhove's lab has also been very productive. He has hundreds of publications, which I cannot mention one by one, but we have one recent publication in 2020 from Nature, that was published in Nature Communications, entitled Common and Distinct Neural Representations of Aversive Somatic and Visceral Stimulation in Healthy Individuals. If you're interested again at all of his list of publications, please have a look at the LaBGAS website. So I think without any further ado, I would like to give the time and screen to Professor Van Oudenhove. Please, Professor, thank you.
Thank you, Dina, for the nice introduction and for inviting me. Delighted to be able to give a lecture to all of you. I would have preferred to come to Indonesia in person. I visited in 2019 and I really love your beautiful country. So I would definitely like to come back as soon as the pandemic situation permits. But in the meantime, luckily, we have modern communication channels and I can still already give a talk to all of you. So thanks for attending on this public holiday. That is really impressive to see so many people here. Happy Islamic New Year for all of you. Happy to be here, and let's get started. Before doing so, I would really like to encourage you to ask questions. Don't be shy, use the chat function. We'll have time for Q&A at the end. Anything you would like to ask, please do. I know it's always a bit more difficult to make things interactive on these online platforms, but still let's aim for it. So this is the opportunity to ask any questions you may have after my talk. So please use the chat function. You can also already use the chat function during my talk, but I would propose to save the Q&A for afterwards. But don't worry if you have any questions also during the talk, type them in the chat right away, and then we'll have a look at them after my talk, and we'll try to address as many as possible within the allocated time. So that being said, let's get started. I'm gonna share my screen here. And all right. Can you see my presentation?
Yes, nice and clear.
And is it in presentation mode? You don't see anything else? You only see the slides? Okay.
Yes.
All right. So the title of my talk is the Microbiota-Gut-Brain Axis between hype and revolution. Hype because it's a very hot topic in medical sciences and psychology and neuroscience, but maybe it's a little bit overhyped, as I will try to explain to you. And otherwise, it might also be revolutionary if some of the early findings get confirmed and translated to humans. So the hype of the day, you see that there's like many papers on the microbiota-gut-brain axis, all of them with quite hyperbolic language, I would say, mind-altering microorganisms, paradigm shift in neuroscience, psychobiotics, a novel class of psychotropics. So there's like big words here. Illustrating how hot this topic is, but also maybe illustrating that it might be too early days to use this strong language when it comes to the microbiota-gut-brain axis in the context of psychology or affective science. One of the signs here of this over-optimism is that if you look at the literature, there's actually more review articles on this topic than original research articles, which is never a good sign, to my opinion. So we should remain critical, and this applies in particular when it comes to human studies, because there's, as I will show you, there's lots of fascinating and spectacular rodent studies, but the number of human translational studies are still limited. The quality is not always that good. And the evidence is also a lot more mixed and complex compared to rodents. So there's also lots of popular books. Here's one by Emmer and Meyer called The Mind-Gut Connection, but there are several others as well. So let's start with the idea that we may only be 10% human. And what I mean by this is that the number of microbial cells in our body outnumber the human cells by a factor of 10, roughly. Well, the numbers are varying a little bit, but they certainly outnumber the human cells. And when you look at the genes, this ratio is actually even lower, the ratio of human genes to microbial genes. So this is a big part of our body. These bacteria are roughly 1.5 kilos of microorganisms we carry in and on our bodies. So maybe it's not surprising that this forgotten organ, as you could call it, plays a more important role in all kinds of bodily functions than we previously thought. So humans are actually superorganisms carrying many other organisms, and you can see, so all body surfaces and cavities are colonized with specific bacterial ecosystems. You see really high numbers, so billions and billions, more bacterial cells than human cells, more bacterial than human genome, biomass of roughly 1.5 kilos, and thousands of species. And as you can see here as well, the composition of the microbiota, which is like this, the collective term for all the microorganisms, it greatly differs between different parts of the body. The most important part of the body, or the part where these bacteria are the most abundant, is the gastrointestinal tract, and particularly the colon or the large intestine, where the majority of these bacteria reside. They like the environment, it's anaerobic, it has a constant temperature, and so on and so forth. You can also see that along the gastrointestinal tract, there's big differences in composition, with the stomach obviously being acidic and differing dramatically from the colon. And also in the stomach, it depends on whether you're H. pylori positive or negative. So we have lots of these bacteria in and on our body, but we also need each other. So as I already alluded to, we provide the bacteria with an anaerobic environment with a constant temperature, and also obviously a stable supply of nutrients, because they can feast on anything that we ingest and reaches the colon or other parts of the gastrointestinal tract. And then vice versa, we also benefit from the microbiota, for example, from their high digestive and metabolic capacity, and from their defense against pathogens or bad bacteria. So this is a symbiotic relationship that has evolved over thousands or probably million years of evolution. Also throughout life, there's variability in our gut microbiota, as you can see. So the unborn child does not have any microbiota. We receive the microbiota from our mother when we are born. That means that whether you're born by C-section or by vaginal delivery, has an impact on your microbiota composition typically. Then in very early life as a baby, breastfed versus formula fed can make a difference. Then things change when we start eating solid food. Then as children or adults, obviously there's factors playing a role, nutrition. Obviously, antibiotics dramatically change the microbiota. And then, of course, in adults, whether you're normal weight or obese will also have an important impact. And then, of course, there's changes with aging, as in any bodily system. So enough about the gut microbiota as such. Now let's talk about the microbiota-gut-brain axis. So what is the microbiota-gut-brain axis? It's basically a collective term for a very complex bidirectional communication system between the gastrointestinal tract, including its microbiota, and the brain. So this goes in both directions. There's many routes of communication that can be roughly divided in two kinds. One of them is neural communication through the autonomic nervous system, afferent and efferent branches, or vagal, sympathetic, obviously. And then there's all kinds of systemic communication routes through the bloodstream, including the HPA axis, which I will come back to later, neurotransmitters, all kinds of bacterial metabolites that may communicate with the brain. I will also come back to that later. And then one of the other important routes is through inflammatory mediators, such as cytokines. So the microbiota have an important capacity to regulate inflammatory processes locally in the gut, but also systemically. So one of these important communication routes is obviously the vagus nerve, which is, as you probably know, innervating all the inner organs up to the ascending colon. Of course, you're all aware of the efferent function of the vagus, which is going to have a stimulating effect on GI motility and digestion, which is going to slow heart rate and so on and so forth. But what people are not always aware of is that 80% of this thick cable that is connecting our brainstem to our internal organs, 80% of this thick, big, massive nerve is afferent fibers. So there's a constant stream of afferent information from the body, including the gastrointestinal tract, to the brain that allows homeostasis. It allows the brain to sense the inner state of the body and to make adjustments, the majority of which are done by reflexes outside conscious awareness. So this is very important. It means that this afferent part of the vagus, which is very important, can also play a key role in microbiota-gut-brain communication. One example of efferent or brain-to-gut communication is the hypothalamo-pituitary-adrenal axis, or the HPA axis, which is the main stress response system, or together with the autonomic nervous system of the human body. So when we are experiencing a stressor, CRF, or corticotropin releasing factor, is produced by the hypothalamus. It then releases ACTH, or adrenocorticotropic hormone, from the pituitary, which is released into the systemic circulation, stimulating the adrenal cortex to produce the stress hormone cortisol. And then cortisol has all kinds of effects, including negative feedback loops. I will turn on my pointer. So you see here, these are the negative feedback loops. So in normal circumstances, there's negative feedback. So the stress response is properly shut off when it's not needed anymore. But cortisol also has effects on immune cells, on inflammation, very importantly, which also feed back or feed forward into the stress system. And also this activation of the HPA axis may impact microbiota composition and function. So let's have a quick look at signaling mechanisms, specifically focusing on the gut microbiota. So imagine that this is the lumen of the gastrointestinal tract. This is the epithelium. So this is the wall of the gastrointestinal tract. There's obviously gut microbiota living here in the lumen and in the mucus layer and so on and so forth. And they basically can affect the brain through systemic and neural routes, as I explained before. So how do they do that? Some sorts of microbiota, some sorts of bacteria produce neurotransmitters like GABA or dopamine or others, which end up in the circulation and which may have an effect on the brain. One important metabolite, which I will zoom in on later because our research focuses on it, are short-chain fatty acids. They're bacterial metabolites that can interfere with all kinds of gut-brain communication, but I will come back to that later. They may also impact metabolism of certain nutrients like amino acids, like tryptophan, which again is going to impact on neurotransmitter synthesis and neurotransmitter degradation. And then there's an important, as I alluded to earlier, immunomodulatory role here, which can impact inflammatory states, immune cells, cytokine production. And again, we know that these directly via the circulation or indirectly via the vagus can impact on the brain in general and the emotional brain in particular. And then vice versa. Again, here you see the HPA axis with cortisol as an end product, having all kinds of effects on the gastrointestinal tract, on its microbiota, and on the immune cells. So you see that it's a very complex bidirectional communication system between the microbiota, the gut, and the brain. So how do we study this microbiota-gut-brain axis? Well, basically the principle, both in rodents and in humans, is that you do an intervention altering the gut microbiota. In rodents, they study these germ-free animals, which are born sterile without microbiota, and then look at their brain and their behavior. I'm not such a huge fan of this because these animals are very different in any way. Their entire physiology is different, so I don't think they're a good model for human microbiota-gut-brain interactions, which is what I'm focusing on and interested in. But there's other interventions which can be used in humans and are less dramatic, including probiotics, prebiotics, dietary interventions, antibiotics, fecal microbiota transplantation, where microbiota from a donor are transplanted in a recipient. And then the effect of these manipulations at the level of the gut microbiota is measured at the level of the brain by looking at subjective responses. Of course, in humans, in rodents, you only can look at behavioral responses, obviously, as a proxy for human emotional states. And then, of course, we can look at the brain, both in rodents, with post-mortem techniques or in vivo imaging techniques. And of course, in humans, we are limited to in vivo imaging techniques, but luckily there's a lot of progress in those, so we can use all kinds of nice brain imaging techniques to look at the brain responses to these interventions at the level of the gut and the gut microbiota. This is one of the things that my lab is actually focusing on. So a few definitions for you to understand these different potential interventions. So probiotics are actually live microorganisms, bacteria typically, which when administered in adequate amounts confer a health benefit on the host. Prebiotics, one letter difference, but a totally different thing, are substrates that are selectively utilized by host microorganisms, conferring a health benefit. So this is these are nutrients, typically fiber, dietary fiber, complex carbohydrates, complex sugars, that are utilized, metabolized by the microorganisms, and then producing metabolites that may confer a health benefit. Psychobiotics is a term coined by John Cryan and Ted Dinnan in Cork, one of the leading groups in this field, good friends of mine, but I'm not a big fan of this term psychobiotics because I think it's too early days. I hope one day we'll be able to use it. But the meaning is either of the above, conferring a health benefit in patients with psychiatric illness. I simply think that at this stage, we don't have convincing evidence that any probiotic or prebiotic may actually be a psychobiotic according to this definition. So that's why I think it's too early days. And then, of course, there's fecal microbiota transplantation, which can also be done and is also being done in humans for one very well established indication, which is Clostridium difficile infection. So this is an opportunistic infection of the colon, which can be life-threatening. And it's typically hard to treat with even last resort, end-of-the-line antibiotics. And there's actually been high-quality clinical trials showing that fecal microbiota transplantation outperforms antibiotic treatment in this particular situation. In more experimental settings, it can also be used, and it's also being tried for various indications and even for mechanistic studies in humans. So let's start to look at the actual research, and we'll start with rodents, and we'll start with one of the papers that opened my eyes. I was pretty skeptical about the whole microbiota-gut-brain idea, but this is one of the papers that really caught my eye and coined my interest and convinced me that there may be something in there here. So this is a paper by the friends in Cork whom I mentioned earlier, back 10 years ago now already, where they basically showed that a probiotic, Lactobacillus rhamnosus JB1 in this case, actually alters emotional behavior and central GABA receptor expression in mice, and that this is mediated through the vagus nerve. So here you see basically the results of 3 stress tests or emotional behavior tests, let's say, in these mice. No significant difference for stress-induced hyperthermia, but the elevated plus MACE, which is an index of anxious behavior, and the forced swim test, which can be treated as an index of depressive behavior, they both show anxiety and depressive behavior reducing effects of the Lactobacillus rhamnosus JB1 treated group. And then if we look at corticosterone response to the first swim test, so corticosterone is the rodent equivalent of cortisol in humans. Then you see basically this is a control group that has not been stressed and is fed with just normal control chow. And here you see the stressed group that was fed normal chow. This is the control group that got Lactobacillus rhamnosus JB1. So in non-stressed circumstances, it's not much of a difference. But you see that the stressed group with Lactobacillus rhamnosus has significantly less pronounced corticosterone response to the first swim test than the stressed group that was fed the normal chow. It's about less than half, so that's pretty strong and significant. Interestingly, also, if they looked at the brain post-mortem in these different animals, so the control fed and the Lactobacillus rhamnosus fed animals, and you basically also see all kinds of differences in regions of the emotional brain in GABA receptor expression, including the amygdala, the locus coeruleus, various hippocampal subregions, and other limbic regions as well. And interestingly, John and Ted and colleagues were able to show that all these effects that I showed to you were actually completely abolished in animals who received a vagotomy prior to receiving treatment. So this shows that you need an intact vagus nerve for these effects to take place. However, and here we're going to touch upon an important part of my talk, which is the lack of human translation. So if you look at translational studies, basically they use the same probiotic in humans and failed to find any effects on stress or cognitive performance, at least in healthy subjects. This is another, I think, very nice study where they had rats or mice, I think, I don't remember, who got antibiotic treatment first. They had 2 groups, a control diet group and a high-fat diet group. So they abolished their microbiota with antibiotics. You see that weight goes down a little bit. And then basically they got a microbiome transplant. And in the end, they got behavioral test. Well, it's basically, they start with the same mice, but they get a microbiome transplant from control or high-fat diet mice. And then in the end, they do a number of behavioral tests. You see that the high-fat diet transplanted animals they become heavier, but the difference is not significant with the control fat diet. So they get basically microbiota from obese animals. They don't become obese themselves, but on behavioral tests, you see all kinds of differences. Again, the elevated plus maze and the open field test, which are tests for anxiety. You see that the high-fat diet transplanted mice, that they become more anxious than the control diet transplanted mice, and that this is independent of overall locomotor activity. So I'm going to skip the details of this task, but basically this is a study in humans with prebiotics, B-GOS, which is galacto-oligosaccharide, and basically they also used fructo-oligosaccharide. In this paper, they showed that the B-GOS arm, but not the FOS arm, compared to placebo, actually dampened the cortisol awakening response, which is a natural increase in cortisol after awakening, which is supposed to be an index of stress. From pre to post treatment. This was treatment for 3 or 4 weeks. They also found effects on another emotional task, reaction time task, measuring emotional bias, but this was also to an important extent driven by changes in placebo. So yeah, I don't know what to think about that. And they did not find differences on a number of other emotional tasks or anxiety or stress related questionnaires. This is a study in humans again using probiotics, showing that it's actually Activia, which may be available in Indonesia. I don't know. It's a well-known probiotic yogurt with a number of probiotic bacteria in there. And this is work from the UCLA group of Emra and Meyer. Also already 8 years ago now, and they basically had a no intervention group. They had a control group with a yogurt without probiotic bacteria, and then FMPP is fermented milk product, which is basically probiotic. So you see that in resting, these are changes in resting brain activity between the probiotic treated group before versus after versus before treatment compared to the control group. And again, they also used an emotional task where they had to label or match affective pictures versus shapes. And also there, you see that in terms of brain responses to this emotional task, there's changes in the probiotic group compared to both the no intervention and the control group in regions that are relevant towards emotions and also processing of bodily signals, particularly the insula, the mid-insula in particular, here as you can see, as well as the somatosensory cortex. So this is a study again using another emotional paradigm, this time in IBS patients, so patients with irritable bowel syndrome with comorbid anxiety or depressive symptoms. And basically what you can see here on top is that in the probiotic, but not the control group, after treatment compared to before treatment, you get significant dampening of amygdala responses to these emotional stimuli in these patients that were absent in the placebo group. There was also an improvement in depressive scores from before to after treatment. And if you look here in the Bifidobacterium longum or the probiotic group, you see that there was a positive correlation between the decrease in amygdala activation and the decrease in depressive scores. So this mechanism at the brain may explain the changes in subjective ratings of, in this case, depressive symptoms. Another way, of course, to look at this is to do case control studies and look at microbiota composition. This is a study, one of the first ones in humans, comparing patients with major depressive disorder or MDD and controls. And you see that there's differences, particularly with higher numbers of actinobacteria and lower of Bacteroidetes in major depressive disorder patients and controls. And you can also see on this PCA plot that both groups can be separated quite well based on their entire microbiota composition. Interestingly, in this paper, they did some reverse translational work where they transplanted microbiota from the depressed patients into germ-free mice. So these are germ-free mice, that the red bars are germ-free mice that received a transplantation from depressed patients, and the white bars are mice that received a transplantation from control, non-depressed patients. And what you basically see here is that on all these tests, again, open field, tail suspension, forced swim tests, the mice that received microbiota transplanted from depressed patients start showing more anxiety and depressive behavior independent of overall locomotor activity compared to mice receiving a transplantation from control patients. This is a large population-based study that I was involved in and that we will build upon further in my moodbugs project. This effort is led by my colleague Jeroen Raas, who is world-renowned when it comes to microbiota studies in humans. He has a big population-based cohort of like thousands of people that are followed up longitudinally with stool samples and all kinds of other measurements. So this is a really nice data set. And we also compared the results with a similar cohort from the Netherlands, the Lifelines cohort from Groningen University. And basically here, this is quality of life score, so mental aspects of quality of life, physical aspects, and self-reported depression. And we basically saw that Faecalibacterium, Coprococcus, and Dialister were associated with better quality of life. So higher abundance of these was associated with better quality of life and with less risk for depression. More generally, if you look at the entire microbiota composition, the people can be divided into 4 enterotypes: Bacteroidetes 1, 2, Prevotella, and Ruminococcus. And on this cohort, we actually managed to show that the B2 enterotype here, that you see that people score lower on all domains of quality of life, and that more people of the depressed people have this enterotype. So there seems to be an association with this B2 enterotype. So what is important here? What do they have in common? They are important producers of short-chain fatty acids, which I mentioned earlier, and the B2 enterotype is known to be a pro-inflammatory enterotype. So this points towards short-chain fatty acids and inflammation as important potential mediators of these relationships. And this is exactly what we will focus on in our moodbugs project. I'm going to skip this for the sake of time. And I would like to conclude with showing a little bit on short-chain fatty acids. So short-chain fatty acids, this is a really nice review article. It's not in press anymore. It's been published 2 years ago and has been cited like 300 times or something since. It's pretty crazy. This is excellent work from my PhD student, Bushra Dalila, who's going to defend her PhD soon. So when we eat prebiotics, dietary fiber, the intestinal microbiota, or some members of the intestinal microbiota are gonna ferment them. We can't digest them, so they end up in the colon. They're metabolized or fermented by these bacteria into, among other, short-chain fatty acids. And as you can see here, these short-chain fatty acids can impact on all the brain-gut communicate or gut-brain communication pathways that I mentioned earlier. Humoral, so systemic through the circulation. It can impact on the vagus. It impacts on gastrointestinal hormones, which have an important impact on the brain, and it has important immunoregulatory functions. So these small molecules are able to interfere with virtually every possible route of gut-brain communication, making them very likely candidate mediators of microbiota-gut-brain relationships, also in humans. And we looked at this in a first-in-human trial with colon-delivered short-chain fatty acids. So we gave people capsules with a low or a high dose of short-chain fatty acids that are only released in the colon thanks to a pH-dependent coating. Kudos to my colleague Christine Verbeke for making this happen. And then we looked at the impact on stress responses. And as you can see here, in both the short-chain fatty acid treated groups, there's a decrease in stress response to an acute stress task from post to pre-intervention, which is absent in placebo, and these were significant differences. So this is first proof of concept that the short-chain fatty acids may indeed be an important mediator of microbiota-gut-brain relationships in humans. And we basically also showed that the magnitude of change in short-chain fatty acid levels was associated with the magnitude of the decrease in cortisol response from pre to post. So the higher the increase in short-chain fatty acids from pre to post, the lower the stronger the decrease in cortisol response from pre to post, pointing towards, again, short-chain fatty acids being the main mediators of these microbiota-gut-brain relationships in humans. I see that I'm sticking well to time, very happy about this. So let me conclude with a few take-home messages. So I hope I've been able to show and convince you that there is intimate bidirectional communication between the gastrointestinal tract and the brain, and that the gut microbiota have recently been identified as a key new player in this communication, that gut microbiota composition and function can be altered by pro and prebiotics and fecal microbiota transfer, and that therefore the microbiota could have a strong impact on our psychological and brain function, but translation from animals to humans has only just started with mixed results, not always as good quality studies. I haven't been able to show you many examples of this, but I did show the effect of the very nice, well, the story of the very nice paper in rodents with the Lactobacillus rhamnosus JB1, which then failed in a translational human trial. So we need more translational human research to establish these relationships more firmly and identify the mechanisms. And this is exactly what I'm aiming to pursue with my mood bugs grant, which was funded by the ERC. So I would like to thank you for your attendance and for listening. I hope you found it interesting. And again, do not hesitate to ask any questions you may have through the chat. We have 15 minutes, so plenty of time for all kinds of questions.
Thank you very much, Professor Lukas, for sharing your knowledge on the current knowledge on the MGB axis, and also of all of the work that you've been doing in your lab. I can see that there already is a question in the chat room.
Great.
So from Alvaro here, if I'm not mistaken, SCFA has been recognized for their potential epigenetic mechanisms. Is it possible that this microbiota-gut-brain axis is also mediated by epigenetic regulation of the brain or nerve cells?
Well, this is actually, I think, a really excellent question in the sense that we are going to look at this. It's part of the Moodbugs project. So one thing indeed that these SCFA do is epigenetic regulation, particularly through inhibition of histone deacetylases. And histone deacetylases are molecules that basically increase or decrease the transcriptional capacity of the DNA. So by inhibiting those, short-chain fatty acids can impact on DNA transcription. So we're going to look at this because we know that short-chain fatty acids can do this. And we also know that this HDAC and this epigenetic regulation is an important mechanism regulating the stress response in stress circuitry in the brain, hypothalamus, amygdala, hippocampus, name it. And we have a way to actually visualize this in humans using positron emission tomography or PET. There are ligands available, so radioactively labeled ligands that are binding to histone deacetylases and various types of histone deacetylases. So we're planning to do a study where we are gonna administer those short-chain fatty acids and then look at whether they indeed impact on HDAC in stress circuitry in the brain. So yes, really good question.
Okay, I hope that answers, Alvaro.
Yeah, if you have any, if it's not clear or if you have any further questions, let me know. Huh?
Okay, are there any other questions?
You can also ask them if you don't want to type them. Now I think it's okay to just ask them.
Yes, you can turn on your mic directly.
Yeah, absolutely.
So here we have another one from Alvin from biomedicine. So how does Synbiotic also affect our gut microbiota and the brain?
Yeah, there's all kinds of new things, postbiotics, symbiotics, all kinds of combinations of probiotics, prebiotics. So symbiotics are combinations of pro and prebiotics. So there's not that much work on them, but of course you could imagine that in an ideal world that they have a synergistic effect, the pre and the probiotic. So that is certainly something that may be promising. Of course, we don't know too much about the interaction between the prebiotic and the probiotic and the effects of that, but potentially they could have a synergistic effect. So I do think that this is definitely something to look further into. And there are studies coming out on symbiotics, but I don't really know of any in the context of the Microbiota-Gut-Brain Axis. But I do think it's a promising option. One of the problems is with probiotics in particular. There's so many different ones that it's hard to tell which one is the right one. And different people may need different probiotics based on their different microbiota composition and so on and so forth. So this is all pretty complex, but I definitely think that in principle, the symbiotics have the potential to combine effects of pro and prebiotics.
Okay, thank you. So there's another question in the chat room. So from Jeffy Gore, I'm curious about the study of gut microbiota in human. If I'm not mistaken, on your slide, it shows that human studies could use antibiotics to observe the effect of changes in gut microbiota to the brain. In this case, wouldn't it create an antibiotic resistant effect, which could give rise to superbugs?
Yeah, that's obviously a good point. It's also, we're planning to do an antibiotic study in the moodbugs project, and the ERC definitely was already asking questions about the ethical aspects of it, to which I answered, and now we will need to apply for approval of the ethical committee. So it's really a good point. The risk is obviously there. You don't want to give antibiotics to people without a good indication. However, I think, yeah, when it comes to like mechanistic studies to really demonstrate a causal effect for the gut microbiota, this is one of the only things we can do that really dramatically changes the microbiota. So our take on this is that we use a non-absorbable antibiotic, which is limited to the gut, where it has local effects, but it does not get absorbed in the circulation. This is on the market for treatment of irritable bowel syndrome, a number of other things. So that's our take on, like, at the same time manipulating the gut microbiota, but minimizing the potential resistance-inducing effects. And we're also not going to give this to like a lot of people. So it's going to be limited to a relatively small sample size.
Yes, okay, so it's definitely something to think about. Okay, another question from Clarissa. How do they control the environment during the human studies you mentioned earlier? Because the microbiota in every individual is different. So wouldn't their diet throughout their life affect the results as well?
Yeah, excellent question. One of the things that we're gonna also pursue is indeed in these mechanistic studies, look at inter-individual differences in microbiota composition, and see whether that is associated with different responses to any of these manipulations. So that's a good point. Diet is, of course, an important determinant. It's hard to control or even measure in humans, but what we do, during the study, we have people keep dietary records. We use dietary diaries, so to speak, to keep an eye on this. And in many cases, we even standardize the diet. If we want to, for instance, do a study where we did a study with a fiber product. There we put all people on a low fiber diet to have them start with like the same kind of diet. We do that a few days before the start of the intervention, throughout the intervention. So yes, good and important point, and a difficult one.
Okay, so we have another question from Kenny. As the gut-brain axis is a bidirectional communication, how significant is the effect of the CNS towards the microbiota, especially in the cases of neurodegenerative diseases that can eventually lead to the anxiety and depression-related problems?
Yeah, good point. I haven't talked too much about the brain-gut direction, mostly about the gut-brain direction, but there is definitely, through the autonomic nervous system and through the HPA axis, influence of the brain on the gut and on the gut microbiota. When it comes to neurodegenerative diseases, that's of course a very complex problem. There's all kinds of theories now that Parkinson's may start in the gut and all that. But when it comes to brain to gut, yeah, of course, these people have different microbiota compositions, but also, yeah, their diet is dramatically altered. They're typically, they may have constipation problems, particularly in Parkinson's, so their transit is altered. So it's very hard to disentangle cause and effect here. But it is definitely a possibility that brain to gut pathways also play a role. It's going to be a circular thing, right? If like gut to brain pathways can increase stress, and then stress can further deteriorate the microbiota, it's always going to be like vicious circle type of phenomena, to my opinion.
Yeah, so very complex indeed. Now we have a question from Edward, who wants to know more about the relation of SCFA and stress response. It was mentioned before that the SCFA level contributes to the stress levels. Do you think this relationship could make it harder to deal with the stress management in patients with IBD due to heterogeneous profiles of microbiota changes in IBD patients?
Yeah. I think this could definitely play a role. There's lots of attention for or more and more attention for like stress and fatigue and psychiatric comorbidity and so on in IBD patients these days. And again, of course, they have altered microbiota, they have a pro-inflammatory state. So these could all play a role. I'm not aware of any studies in IBD looking at short-chain fatty acids and their impact on stress, but I could imagine that because of the altered microbiota composition, the pro-inflammatory state, and actually the B2-like enterotype, there's another publication by Jeroen Raas group showing that the B2 enterotype is not only linked to depression, but also to IBD and other inflammatory disorders. So yes, I do think these phenomena could definitely play a role in IBD and in let's say psychiatric comorbidity or stress and fatigue in IBD. Very good point.
Okay, now from Joanne Nadia. I'm wondering whether the common saying happy gut, happy brain is true, considering different enterotypes and that certain bacteria species are indeed related to better quality of life.
Yeah, I think it's true. I think you can use that saying. Of course, it's a saying, but I think the gist of it is probably true because the B2 enterotype, you could see that as an unhappy gut enterotype because it's a pro-inflammatory type of enterotype. So yeah, you could conceptualize unhappy gut as the B2 enterotype, and we see that this is also associated with a more unhappy brain, so to speak. So yes, I think it could apply. Yeah, there's many of these sayings that are alluding to gut-brain connections in many languages. Simply things like butterflies in the stomach and all that kind of stuff. So there's lots of these expressions, and I think there's something about them.
Okay. Now from Teresia. So she has found and read a paper regarding the gut microbiome and their effect in the human intelligence. And it's already undergoing human trials. So she's wondering if the genetic and microbiome composition can affect someone's intelligence. If so, is it possible to change someone's diet for intelligence purpose?
Wow, that would be big. I'm not aware of this for now. Maybe they are doing human trials. I don't think. I mean, of course, there's genetic aspects to intelligence. I don't consider it completely inconceivable that the microbiota or the metagenome, as it is called sometimes, could also play a role when it comes to regulation of intelligence, but I would expect this to be a developmental thing, right? Which takes place in like probably early childhood or throughout childhood. I would find it very, yeah, I would not expect that you could change intelligence in adults still. Because it's such a developmental thing. What does not imply that you maybe could not change like concentration or attentional functions or stuff like that. But intelligence, I would say maybe in a developmental context in adults, it would surprise me.
Okay, I guess that would be the dream, right?
Yeah, that would be fun. Yeah.
Okay, so it is actually already 3 p.m., but we have one last question, if that's okay, from Davin Safiro. So he recently did a metabolic modeling study for soil microbiome community and found that there is an extent of similarity in functional capacity between microbial members that are taxonomically close in terms of class. Are there similar associations within the gut microbiota in which taxonomically close members may have similar roles in affecting the gut-brain axis? So it might be possible to confer similarity in different patients based on the grouping of members, even if the species or genus are different.
That's a really interesting question. It unfortunately goes a little bit beyond my expertise. I think this is more something for a microbiologist. This would be something for my colleague Jeroen Raas or someone from his lab. But I would encourage, from a Microbiota-Gut-Brain Axis perspective, I would encourage you to read this Nature Microbiology paper that I showed you, where I showed the work on the association with depression and quality of life. But there's a big part that is focusing on what we called gut-brain modules. In other words, that based on genetics, the metabolic potential of different genera or species to produce or metabolize neuroactive compounds. So that is work that is, yeah, along the lines of what you are alluding to. So I think it would be good to have a closer look at that.
Okay, thank you. Well, there actually is one more question. Would that be okay?
Yeah, sure.
Okay, maybe. So everyone, hopefully this will be the last question. Sorry, we have very limited time. So Albert, I found that herbal supplementation can increase the SCFA, for example, curcumin compound from Sedwari turmeric shows a positive effect by regulating intestinal flora, resulting in butyrate and propionate production. Can we use this herbal supplementation approach to improve the gut-brain microbiota potential?
Well, I think there's definitely room for this kinds of herbal supplementations too. I know that there's a lot going on about turmeric and curcumin. I'm not aware about the exact details of it, but as long as it is tested in good studies and it's shown to increase the butyrate and propionate production in sufficient amounts, it is a promising thing to look into. Yeah. There's a lot of potential, I think, in these herbal medicine approaches. Many of them are not very well studied, but I think we should do more proper studies on these and then maybe build a better evidence base to also use them. So yes, I think this is a promising route.
All right. Thank you very much, Professor Lukas, for your time and for your talk. Unfortunately, okay. Sorry, before we close the talk for today, we have a reminder here from Ms. Weirda for everyone in the audience to please fill in this evaluation survey in order for us to review and always improve our power talk. And also those of you who fill this in and registered will be given a certificate of attendance. So on behalf of I3L, I would like to thank Professor Van Oudenhove for the very insightful talk. Hopefully sometime soon we can actually have you in I3L physically to give another talk probably.
Really looking forward to that.
Okay, so with this, I would like to end today's Power Talk. Thank you very much for the audience for listening and joining our talk. Have a nice day, everyone, and stay healthy and safe at home. Thank you.
Thanks for having me, and thanks for all the great questions. Enjoy your holiday.
Thank you.
Bye.
Bye.